My name is Sarah Widener Richards, and I'm with the Alexander Lipka Lab at the University of Illinois. And today I'll be talking about perennial sorghum. So the problem that we're facing is food insecurity is really high, and especially in certain regions of the world. One of these regions is sub-Saharan Africa, where population growth is really high, and climate change is felt very severely. One potential solution to help alleviate some of this food insecurity is perennial sorghum, which is a drought-resistant crop. Sorghum is a crop that's readily consumed in this region of the world. Right now, it's an annual system, meaning it's planted every year and harvested once a year. In a perennial system, you plant once a year and then you harvest for multiple years. Uh, yeah. Our goal is to develop a multi-environment genomic selection model for faster breeding. So we're trying to create a tool for breeders so that they can make this perennial sorghum able to compete with annual sorghum really fast. And we have two objectives. One is to optimize models for multiple environments, and the other one is to evaluate impact of environmental data in limited sample sizes. So this is sorghum bicolor. It is what is grown in most of the world. Whenever you think of sorghum or whenever you buy sorghum at the store, maybe in a gluten-free flour mix, this is what you're consuming. It looks a lot like a maize plant or a corn plant. Has really nice big leaves, and the panicle or the seed head has a lot of really large, tight seeds that aren't shattering or falling all over the place. It's nice and cupped in, and that is all domestication traits that we are looking for. This is sorghum helipens. It is the opposite of that. It's actually a weed in much of the world. It has these tiny leaves. It's like scattered all over the place with lots of stems. Its seed head is open. As you can see, there's really small little seeds that are just sporadically placed. And these two get crossed together and they make this succession, which is a blend between the two. We can see some domestication traits, so some bigger seeds and larger leaves, but there's still a lot of weediness traits also. So the population that we're looking at is sorghum bicolor crossed with sorghum helipens, and this created two populations, the H4 and the H6. And then these were back crossed with sorghum bicolor again to try to integrate more of those domestication traits. And then these were genotyped and phenotyped in 2013, and again phenotyped in 2014. We have a really small sample size. Normally, whenever you're doing plant breeding like this, you have thousands and thousands of accessions. In our H4 population, we have 146. In our H4 population, and in our H6 population, we have 108. We also have a really small amount of genetic information. In our H4 population, we only have 722 SNPs. In our H6 population, we have 795. So these plants were grown in Salina, Kansas, and Athens, Georgia in the years 2013 and 14. And now we're moving on to models. So I'm in a statistics lab. We do statistics. We get the data from other people who do a great job collecting it, and we see if we can make sense of it and make predictions. So our phenotypic bluff, which is what we think the phenotype will be at a specific genotype in a specific environment, is the grand mean plus the genomic effect, so the effect of the genes, plus an error term, which is what we can't explain. And we expect a basic model like this to have a very large error term because we're not explaining a lot of it unless the genomic information is really strong and there's not a large environmental factor. So we ran this model, and then we ran a multi-kernel model. And a good way to think about kernels is just as bins or just places where you separate data. So here we have our phenotypic bluff is equivalent to our grand mean plus genomic effects 1 and 2 plus our error term. And what genomic effects 1 and 2 are is we ran a GWAS, and the GWAS identified some peak SNPs or regions of the genome where there's a high correlation to our trait of interest in one population, and then we found that same section in the second population, and that became our genomic effect 1. And then the rest of the genetic information went into genomic effect 2. And the reason that we did this, the core idea is that these peak SNPs were potentially blocking us from seeing some of the medium and small SNPs that still had a significant contribution. So we tried this model, and we did it in real and simulated data. In real data, we found that GBLUP actually outperformed multi-BLUP. And then in our simulated data, when we had 5 QTNs, multi-BLUP tended to outperform GBLUP. So when there's very few QTNs that control a trait or regions in the gene. With 15 QTNs, they perform about the same, and with 30 QTNs, multi-BLUP was outperformed by GBLUP. So in most instances, also multi-BLUP had a larger spread of response than GBLUP, making it less reliable. Overall, the main takeaway from this slide is perenniality and domestication traits are very complex and controlled by a large number of QTNs traditionally. And so we can use GBLUP moving forward, which is a nice result because it means we can keep our models simpler. Next model is G by E with no environmental covariates. I'm going to go ahead and explain what an environmental covariate is. It's a little bit of information that explains the environment at a specific time point. So for us, we did every day minimum temperature, the maximum temperature, and the precipitation or rainfall and snowfall. And we put all that information into a large matrix and included it in our model. But we did not do that with this one. For this one, we took our phenotypic bluff and we put our grand is equivalent to our grand mean plus the genomic effect plus our environmental effect, so the effect of the environment on the plant, plus the genotype by environment effect, which is how the genotype interacts specifically with the environment, and then there's an error term. Next, we have G by E with environmental covariates. So this piece of W term is where we're including that environmental covariate information. So our phenotypic plot is the equivalent to our ground mean plus our genomic effect plus our environmental effect plus our G by E interaction effect plus our error term. And then in order to see how well these models do, we do a cross-validation scheme. And the one that we chose to do was CV00. And this is important because not all of the target environments are included in the training set, nor is all of the germplasm included. So here's some of the results for the traits flowering 50%. So whenever a plot reaches 50% flowering and plant height, these have the highest predictive abilities. They have nice short box plots, meaning less variability of response to our models. Our x-axes are our year location combinations or our environments, and our y-axes are our predictive abilities, and then the different colors are our different models. Branching and tillering were really hard to predict. These are perenniality traits. As you can see, sometimes they're even negative. X-axis is the location year combination, and y-axis is predictive abilities. On the left, we have tillering. On the right, we have branching. On the left is model 3. On the right is model 4. Here is a conglomeration of all of the traits and models on the x-axis and the predictability on the y-axis, and the environments are the different colors. And from this crazy collection of box plots, what I want you to take away is that perenniality traits tend to have lower predictability than traits related to above ground phenotypes. So underground information is really hard to project what's happening there versus above ground information. And there's a couple of reasons this might be. The two main ones are all the weather information that we're including in our models is above ground. So we're looking at air temperature and precipitation above ground. Maybe we could look at soil moisture and soil temperature. And then the other thing is underground traits are really hard to phenotype accurately. And so the more phenotyping information we get and the more tools we get to phenotype really well, maybe high throughput phenotyping tools, the better our predictive models will hopefully be. I'd like to thank my thesis committee, lab members, coaches and mentors, and funders. Thank you.